Again, we see our two different types of reactions that we have introduced earlier. We had dehydration synthesis versus hydrolysis. And of course, when we are building something, when we're putting the fatty acids on the glycerol, we do that by dehydrating or removing water, so we open up the bonds. So we go from the 3 fatty acids and glycerol to the triglyceride at the bottom. And that's how we store the fat. But if we want to use the fat that we've stored, then we have to do the opposite. We have to do a hydrolysis reaction. So starting at the bottom, we'll take a triglyceride. We actually add water to it. And with energy and enzymes, what we do is we split the water so we can actually split the molecules. So we'll add a hydroxide group to one to the fatty acids. We'll add a hydrogen to the glycerol areas, and that will basically split the molecule because we fulfilled the octet rule by adding the parts of water to it. Again, hydro is water, lysis is split. So we split the water to split the molecules. So starting at the bottom and going up to the top, we see that we actually get the fatty acids. Now another thing, notice the top 2 fatty acids are saturated and the third fatty acid is unsaturated. And the difference in that, we'll look at very briefly, is that the unsaturated always has a double bond between carbons within the molecule itself. And so when we say something's saturated, since it doesn't have a double bond, then that means it has room for a hydrogen. Whereas when we have a double bond, we don't have as much room for the hydrogen, so we are not as saturated with hydrogen, so we are unsaturated. When we don't have the double bonds, then we are saturated. We're saturated with the hydrogens. Again, looking at the top, is a saturated fatty acid, no double bonds. The bottom fatty acid is actually a unsaturated. In this case, it's just one double bond. It is a monounsaturated fatty acid. Now, the very ends of these, there's a CH3 on both ends, but there's also what's called a carboxylic acid group on the extreme right end. We're not really concerned with the double bond there, but I do want you to realize that is what's called carboxylic acid, because we'll actually see that as being the acid that makes amino acids an acid. So this is a fatty acid, has a carboxylic acid group on the end. The top one is saturated because it has more hydrogens. The bottom is unsaturated because of the double bond has less hydrogens. Here is an example of cholesterol. Notice the ring structure, which is basically common in all cholesterol-based products such as estrogen and testosterone. Cholesterol, again, is one of those important molecules for the body. We tend to have a bad name for it because people tend to get too much because of their diet or habits. But really, cholesterol is one of those things that is necessary, just like air is necessary in the tire, but you can get too much or too little. Again, here, notice the common theme in all of these structures. Notice that the ring structure is similar in all of them because they're all based on cholesterol. Top molecule being cholesterol, and then the 2 hormones, which are similar but not the same. Obviously, they're doing different jobs, one for female characteristics, one for more male characteristics, even though males obviously have a lot more testosterone than females. Females have more estrogen than males, so these hormones do determine those characteristics. But notice that they both have the same ring structure of cholesterol, meaning they are derived from cholesterol. Obviously, we need all of the organic molecules in our cells, but by far the most important organic molecule would be our proteins. And the building block of the protein are amino acids, which we'll look at here in a few minutes. Now, the building block of carbohydrates would be sugars. The building block of our neutral fats would be the fatty acids and the glycerol. And the building blocks of our proteins are amino acids. Since proteins and amino acids are organic, then they obviously have carbon, hydrogen, and oxygen. But now one more element is also seen in every amino acid or protein, and that's going to be nitrogen. And then sometimes we'll have sulfur in what's called the R groups. But every amino acid, the building block of every protein, is going to have carbon, hydrogen, oxygen, and nitrogen. Now, the proteins actually make up all of our, what they call here, construction materials. They make up all the important structures that are occurring in the cells, or fibers and connective tissue, or just structures that have certain jobs in the cells, such as in a red blood cell, hemoglobin is the protein that would allow it to do its job. In muscle, proteins such as actin and myosin, troponin and tropomyosin would actually make up the part of the muscle that actually contracts, which is the majority of the muscle. So the things like keratin or collagen, there's many different structural proteins found in different types of cells. So it's very important for cell functions. It's also very important that all enzymes which make the chemistry possible within the cell, and many hormones, not all, but many hormones, and all antibodies are actually made up of proteins. Now, this is just a very small list of what's actually made up of proteins, but it'll give you an idea that proteins really are the most important organic molecule in the cell. Here we see an amino acid, the building block of all proteins, and so let's see what all amino acids have in common. Now, there are 20 different amino acids, but this picture shows what they all have in common. Starting in the middle, we see that there's something called a central carbon. It has 4 bonds to it. To the top is a hydrogen bond. To the pictures, or looking to your left, we see the amino group, which is made up of nitrogen and hydrogen. And then on the right-hand side, we see the carboxylic acid group, which is basically the same group we saw on the end of a fatty acid. So since we have an amino group on one side, a carboxylic acid group, that's where we get our terms amino acid. Last but not least, we see the R group, and what the R group is, the R group can be 20 different things that would be attached there that have different properties. So the R group is what makes one amino acid different from another. Okay, now here we see 2 amino acids that are getting ready to bond. And of course, when we put stuff together, we do it by dehydrating what we're putting together. So notice that we're going to take the hydroxide from the carboxylic acid side of the glycine. We're going to take the hydrogen from the amino side of the alanine and combine those so we can open up the bonding spots between the nitrogen and the carbon. And of course, we call this a peptide bond. Now again, notice glycine and alanine have the similar structure of every amino acid. They have the amino group, they have the central carbon, they have the hydrogen, they have the carboxylic acid group, but they differ in the R group. For glycine, the R group is simply a hydrogen atom. For alanine, the R group is a CH3 group. So we put these together, we form what's called, in this case, a dipeptide, because there's just 2 amino acids. But when we eat something, when we eat a protein, then what we have to do is actually break it apart, again, using energy and enzymes. But what we've got to do, according to this, is do a hydrolysis reaction. Because if I want to break apart that dipeptide, I have to add water to it, but I need to split the water so I can split the peptide bond. And when I split the water, I put the hydroxide group back on the carbon so I can complete the carboxylic acid. And I put the hydrogen back on the amino and the nitrogen so I can complete the amino group. So again, we see the same dehydration synthesis and hydrolysis. Again, if I'm putting something together, I dehydrate it to open up the bonds. If I'm breaking it apart, I add water to it, split the water so I can split the molecule. Now, here we see actually all 20 amino acids, and we see what they all have in common. They all have the amino group, central carbon, the hydrogen, the carboxylic acid. But what we see highlighted in each of these is the R groups, and the R groups are what make one amino acid different from another one. Now, notice that we put these in different categories into nonpolar, polar, uncharged, and polar charged. And so the important thing about that is that the order of the amino acids in making the protein will determine their attraction for each other, the attraction for the water around them, And ultimately, what it will determine is the shape of the protein. And the shape and the charge on the protein really determine what the protein can do. Kind of like you might have different keys, but they all have different shapes because they can all open different locks that require different shapes. So in the body, shape is everything. In the protein especially, shape is everything. What determines the shape is the number of amino acids, the order of those amino acids, how those amino acids interact with each other, and that is determined by their R group and by the condition of the R group, such as is it polar charged, polar uncharged, or nonpolar. Enzymes are extremely important proteins that basically allow chemical reactions to proceed at the energy level and speed that they need to occur. Now, I'll be showing you a little video later that really will explain this in a little bit greater detail and increase your understanding. For right now, though, we know that we want to look at the fact that we have these enzymes at certain shapes, and those shapes fit with the chemical that are going to go through the reaction so that they will go through the reaction correctly and at the energy level that they need. Now, sometimes different things in our body can actually change the shape of that enzyme by breaking bonds within the R groups that are holding the enzymes in a specific shape. When that does, we say we've denatured the enzyme, and that causes it to quit doing its job. So each cell has the potential to have up to 2,000 different enzymes, usually not all at the same time. And enzymes are very specific for specific chemical reactions, but they can be used over and over. Our last group of organic molecules are the nucleic acids, and these include things like DNA, RNA, and ATP. And the building blocks for nucleic acids are called nucleotides. Nucleotides have a phosphate side and a sugar, either ribose or deoxyribose, according to which molecule it is. And they also have something called nucleotide bases. Now, there are 5 nucleotide bases, but only 4 of them would be in DNA and 4 of them would be in RNA. Adenine, guanine, cytosine, and thymine would be in DNA. Adenine, guanine, cytosine, and uracil would be in RNA. Here we actually see some DNA, which is a double-sided double helix that actually the sides again are made up of the sugar phosphate backbone. The bases are made up of either thymine, which only bonds to adenine, or cytosine, which only bonds to guanine. So what DNA does is it contains our genes. What our genes do is they actually give us instructions of how to build all of our polypeptides and proteins. Our major energy source for the cell is adenosine triphosphate, we usually call it ATP. And this is something that is stored in the cell and that is replenished by breaking down glucose and actually getting the electrons and the hydrogen atoms out of glucose and generating this particular energy source in a process called cellular respiration, which we will be discussing in the near future. Now, the energy is actually contained between 2 phosphate groups, and we have to break off one of the phosphate groups in order to release that energy. When we break off that phosphate group, we say we are phosphorylating something. But ATP, again, is super important. Our body will use all sorts of things to get ATP, and if it doesn't have glucose available, it'll use other sugars, or it will even use the fatty acids off of triglyceride, or it can even use an amino acid off of a protein. But the main thing is it's going to try to reproduce this ATP by adding the phosphate back to the adenosine diphosphate, which is what's produced after we break it off. Here we see an adenosine triphosphate molecule. Notice we have adenine and a ribose sugar makes up the adenosine. Then on the left side, notice we have 3 phosphate groups. Now, the energy is actually stored between the oxygens that are on the different phosphate groups. So if we break that bond between the phosphate groups, we release that energy.